So, number 1 says, you should be able to explain and label the basic structure of viruses.
Capsid.
Capsid?
Can you draw one?
Some have envelopes.
DNA
Not
or
all.
RNA.
Yeah, genetic material. So there's genetic material, either the DNA or RNA, depending on what type of virus it is. And then there's the capsid. Some viruses will have envelopes.
Nice, that's good. Yeah, okay.
Genetic material. Some viruses will have accessory structures and things like that. But when it comes to basic structures of viruses, those two things.
So if you were to label them, the capsid would be on the outside and the genetic material
So
would
it's
be on
just
the
two
inside.
things?
Yeah.
That's how.
So that's all we need to actually know.
For the overall, the basic structure, yes.
So we have to explain that to them.
Yeah.
Well,
Capsid.
yeah.
What it is.
Explain, there might be a question like, you know, what is the basic, describe the basic structure of a virus. So you just have to say, okay, it has a capsid around the outside to protect it. It has genetic material, either DNA or RNA on the inside. Huh? Okay, number 2, you should be able to identify the basic types of viruses. So, what were the types that we talked about?
CLCC.
What?
No.
Are you recording this?
Yes. You think you guys? So far, I've heard bacteria types. We want virus types.
Bacteriophage.
Bacteriophage. Okay, what else? Enveloped. What else? Helical.
Caucus is a spiritual shape.
No, that's bacteria. One more. Polyhedral.
Yeah, but
Hey, you want to go back to your picture back and then look at those?
Oh, because you might have to label them based on how they look.
Perhaps.
What's the one before polyhedral?
Enveloped.
No. After.
Helical.
Oh, sorry. Yeah, helical. Well, enveloped is before polyhedral. It's just a little farther before. So those are the four main types. And yeah, look at the pictures. Be able to recognize them. Okay, on the test, Cedric,
Goodness.
you will have to be able to explain factors that support and negate the idea of viruses being living organisms. So, what type of things would we say in favor of viruses being living organisms? Bryce?
They can metabolize. Have metabolism. No, they don't have
Not
metabolism.
really.
oh my god.
Do they not have metabolism?
Not
They
really,
evolve.
not really.
Right. They evolve.
Jada?
They can adapt to their environment.
Okay, they can adapt to their environment.
They can have babies
They can
in a host cell.
We want to talk about the things that are going to be supporting the idea that viruses are alive. They can adapt to their environment. A bunch of people have already said they can evolve. Okay. Yes?
Grow and develop.
Yeah,
Supported, non-regular.
that one's sort of, but the other main one for
Structures.
oh
Yeah, non-regular.
is the non-random structure. They have a specific structure to them, and we see that by looking at the pictures, and we can tell the different ones apart.
So there's 3 there?
Yeah, those are the main ones. Okay, some of those other ones like grow and develop, you gotta have a host cell do that for them. Okay, what things would we say in support of denying viruses that were meant to be alive?
Homocells and reproduce both.
Okay, reproduction and growing and development take a host cell. They can't do it by themselves. And they also can't really do what, Bryce?
Have babies.
Metabolite cells.
Asked you specifically because that's the one thing that you came up with.
Well, I thought I wrote yes by that.
I did too. We don't kind of.
Kind of.
And can't they not do homeostasis?
Right. The other main one is they do not have homeostasis. They don't have any way of controlling their internal environment. I mean, even bacteria, because they have a cell wall that helps them regulate the pressure that's on the inside. Now, it's passive. It's not like they're actively doing things. But they have a way of keeping themselves from blowing up. Bacteria don't really have, or viruses don't really have anything like that.
So they just don't blow up circulation.
They don't blow up because they don't have, the capsid doesn't let water through, so they don't have osmosis trying to. Okay. All right, number 4, you should be able to explain and label the basic parts of the Leydig cycle. So remember, the Leydig cycle is the most simple one. That virus attaches, gets its genetic material inside the cell, makes the cell make copies, they erupt from the cell and infect other ones.
You should write.
Or repeat slower.
How's that?
That's her right.
I mean, if I tried to write all that up there, you're not going to be able to read it. So the viruses attach to a cell and get their genetic material inside. Then remember, the main part is they are going to kind of take over the cell and have the cell make more of themselves.
Oh, that's all.
Or two, I mean, I don't know.
Well, it's a cycle. I give it a cycle.
Yeah, it's a cycle.
So it doesn't really.
Depends on where you start. And then, once there's more of those viruses, once the cell builds lots of those viruses, they're going to erupt from the cell. They're going to kill the cell and destroy it and break out. And then they can go infect other cells.
Is that the most common type of virus?
Well, all viruses go through a portion of the Leydig cycle. When we talk about the lysogenic cycle, that's kind of an add-on effect. For the viruses to actually make more viruses, they'll go through the Leydig cycle too.
I just sneeze, but it
Okay? Okay, number 5, the lysogenic cycle. Remember, that's the one where the virus, instead of making the cell make more of itself, it just kind of gets its DNA or its RNA in there and lets it hang out. And the whole point of the lysogenic cycle is that when the cell divides, then the virus DNA / RNA gets copied. Now, we talked about how in cold sores the virus will spend time in the lysogenic cycle. As your cells copy themselves, they're making copies of that DNA. And then something can set it off and trigger it to go into the Leydig cycle. So it's not like the lysogenic cycle is completely separate, like it's an alternative. It's just kind of another thing that some viruses can do to kind of hide in the host cells for a while before they go into the Leydig cycle. So they're not separate things. Some viruses only go through the Leydig cycle. Some will spend some time in the lysogenic cycle and then go through the Leydig cycle.
Which one usually happens first?
Normally first.
Oh.
They're all cycles.
Yeah.
I mean, that you have to go back to the, chicken and egg thing. When was the first thing developed? Okay, number 6, prions. Misfolded proteins. So, what's so dangerous about a misfolded protein?
They may affect other proteins.
They can affect other proteins and make them misfold.
Is this the E or the A? Does that affect your A?
Oh. They can affect, a change. Okay? So then that's the basics of it. This folded protein gets inside you.
You're screwed.
Basically.
It bumps into other proteins, causes them to misfold in the same way. So it acts like a disease that way.
So how does it cause disease, really?
Because it's when it causes other proteins to misfold, those proteins aren't doing what they're supposed to in your cells, and that causes the cell to die. Okay. Remember those? That's the thing we talked about with bovine, the cow, mad cow disease, where it creates the holes in the brain because your brain cells are dying. Proteins aren't working right. All right, number 7. Explain how bacteria cells compare to the cells of eukaryotes. So this is kind of a throwback. It's review. But we should know this. What is the main difference between bacteria cells and eukaryotic cells? Like the definitional difference. What? Right. No nucleus in the prokaryotic bacterial cell. Now, you guys are right. The cell wall is slightly different in a bacteria cell, but there are eukaryotic cells that have cell walls. Fungi and plants both have cell walls. They're different types of cell walls, but they do have cell walls. So the main difference between bacteria cells and eukaryotes, no nucleus. They also don't have things like Golgi apparatus and some of those more complex mitochondria, those complex organelles. The main difference, nucleus. Okay, here's the moment you guys have been waiting for. What are the different types of bacteria? August. What do those look like? Spheres. What's another one?
Spirillum.
Spirillum. What do those look like? Spirals. Bacillus. And they are rods. So you got 4 different types of bacteria, or virus shapes, 3 different types of bacterial shapes. You got to know the differences between those. If I show you a bacterial cell, don't call it a polyhedral, because those are viruses.
Polyhedral look the same.
Polyhedral do not look like any type of bacteria. Okay, number 9. What is bacterial conjugation, and what is the point of it?
To like share
Things.
It's so
Toys. I'm gonna trade Pokemon cards.
Oh yeah. They didn't trade those.
Okay, yeah, they're sharing genetic material. Why? Why would you want to share genetic material?
So you don't go
Because it's fun. No, so that you have genetic diversity.
So they can destroy you.
Wow.
That's not their goal.
Yeah, remember, bacteria are not out to destroy stuff, they're just living. Every once in a while, one will cause an infection because it gets into a place where it doesn't normally live. It's What is the structure that bacteria use for conjugation?
No.
Phyllis.
Right.
Phyllis.
I said
Iris.
Okay, whatever.
Yes.
It's loud. I said Phyllis.
Let's write that one down. The plural is pili, P-I-L-I. Pilus extends, puts into the other one. Genetic material is passed. Hey, what is an endospore? Where would you find it? What's its purpose?
It's like a dormant virus.
Okay.
Not viruses.
It's bacteria
Right.
cell.
They form around dormant bacteria cells. What's the point of it, Katie?
It's highly resistant to environmental stress.
Okay, highly resistant to environmental stress. Basically, it's an extra protection layer. When a bacteria finds itself in an environment that it doesn't live very well in, it can create an endospore, just kind of hide out until the environment returns to the way it wants it to be. Bacteria that can form endospores have been able to survive to experiment in outer space, come back and be just normal the way they were before they formed the endospores. So it's kind of like a suspended animation sort of thing. Suspended animation.
No.
Like in some of those sci-fi movies where they're like, here you go in this take, and you won't age for the next 40 years while the spaceship goes across.
So it's like cap on ice.
Yeah, basically the same thing.
Like Cap on Ice. Captain America.
Oh. Can you see that movie tomorrow? I don't understand.
I could see that movie tomorrow too.
Spider-Man Walker. Spider-Man.
Who wins? Don't
Spider-Man. All right.
That's what happened to kill Iron Man.
We are focusing on bacteria.
She said Captain America.
Okay. Now listen. Gram staining. You need to know what color means which.
Purple is gram plus light.
Gram positive, right?
Positive.
Light pink is gram
Negative.
negative. Why does that matter?
Because it tells you what kind of
Different
antibiotics
antibiotics.
and what antibiotics you use.
Right. What antibiotics will be useful? If you watch some of those medical dramas, like ER is not anymore, but
Grey's Anatomy.
Grey's Anatomy, any of those. If you watch them and you listen along, every once in a while you'll hear them talk about gram positive or gram negative bacteria, what kind of regimen they're going to use as far as antibiotics go.
Is dark purple positive?
Dark purple is positive. Light pink is negative. So remember, they take this thing that it's a stain, it's a dye, they spread it across the bacteria, they kind of wash them off. If they're stained dark purple, they're one type. If they're light pink, they're the other.
Is that all we need to know? Oh, and then water.
But what? Yeah, why you would ever want to do that? Figure it out. Okay, number 12. The difference between anaerobic and aerobic bacteria. This is another way to kind of classify bacteria. What is anaerobic?
No oxygen.
Without oxygen. Aerobic means they take oxygen. Now listen, we talked about how some anaerobic bacteria will actually die if they're in an oxygen environment. Don't think that that's the definition though. Not all anaerobic bacteria will die in an oxygen environment. They just don't need oxygen. They're perfectly happy working without any oxygen around. Some of them will get killed by it, but that's not the definition. Aerobic bacteria, though, have to have oxygen.
That's a need.
Where would you find anaerobic bacteria?
Bottom of the ocean.
In your
That would be a
stomach.
place, but also in your stomach. You don't have to go all the way to the bottom of the ocean. Right there, you got some.
Don't even have to go that far. Grab a napkin.
Yeah, it doesn't hurt. Do it.
Yeah, don't do that.
I don't do that. No, I could never do it. I'd be like,
I'll do it.
I'll do it for you.
Okay, number 13. What are the two main ways in which bacteria cause disease?
They break cells.
Oh, there's
They metabolize their host,
or they release toxins.
or they release toxins. They eat you, or they poison you.
Wow.
what is the most common thing as far as bacteria that affect humans? The toxins. Even the so-called flesh-eating bacteria don't actually eat you. You guys saw that yesterday, right?
Aquarius, whatever.
Yeah, that was pretty gross.
Exciting fasciitis. Yeah, like I said, I don't know if any of you went out and Googled after that, but that is like the nicest picture that you could find.
Yeah, Katie.
So, just remember that. They either metabolize their host, use you for energy, or they produce toxins, which is their overall byproducts, and those toxins.
We didn't have that picture.
No, I added a couple pictures. Sorry.
Didn't want to take her.
Okay. But from the videos that you watched yesterday, what are some beneficial uses of
Break down
bacteria?
your food.
Yeah, break down the food in our gut. That's one. What else? You actually pay attention to the videos yesterday?
Yeah.
Yeah.
They make your breast smell bad, but that's
Well,
Yeah,
that's
to
not-
talk about your food falling on the floor. He needs.
Okay, we did that.
Oh my gosh.
That guy.
Frozen sand on the floor.
Right.
Yeah, we ate it.
Talk
That's
about
disgusting.
beneficial. So,
Um,
the fermented foods.
I asked those food.
Okay. So, certain types of sauerkraut, right?
Nasty.
That's fermented food. Bacteria are working to make sauerkraut.
So, a benefit is they make us food.
Yeah. Cheese is, if you've ever eaten cheese, that's a bacteria. You can almost classify cheese as its own living organism. There's so many bacteria in it. They are harmless,
Sugar.
but that's what makes the cheese from the milk, from the whey.
So what were you right there?
The benefits is great food and big food.
Right. Everywhere, cheese and fermented foods. Also, remember, they can produce alcohol.
Oh, what a benefit. In fact, the same thing. People in
Exactly.
Well, you know,
So, listen. As I said yesterday, most alcohol is made from yeast, which is not bacteria, but there are certain types of alcohol that are made from bacterial strains instead of yeast. Okay, and then finally, antibiotics. How do antibiotics work?
They ruin the,
So long, and
yeah, and then water.
Right, the water that's continuously rushing in, always rushing in, then can build up enough pressure to make the bacteria explode. Don't get the idea that the antibiotics create holes for the water to come in. The cell wall is holy. It's more like a net. So water gets in no matter what. The whole point is that it's rigid enough that it keeps the bacteria from exploding under normal conditions. What the cell wall does is basically, the antibiotics basically do, it can't hold its structure. But what are the effects of overuse of antibiotics?
Mutated bacteria.
resistance.
Yeah, we
Superbugs.
basically force evolution on the bacteria, and the resistant ones live, and that's all the type of bacteria there are, and then the antibiotics don't work. So, here's the thing. They have done surveys with doctors, medical professionals, listen, who basically have said, if somebody comes in and demands antibiotics, they usually just write it up. Because it's a lot easier just to do that. That's what makes the person happy. They know it's not going to do anything for them, but the person is convinced they need antibiotics, so they give it to them. And the problem with that is, it's not helping, so all it's doing is fostering this antibiotic resistance. So basically just listen to your doctor. Don't demand antibiotics. If it's not a bacterial infection, antibiotics won't help you. No use in taking them. All you're going to do is add the problem.
Don't be done.
Okay. I am weary. I think we're done.
Weary.